All right, good evening, everyone. My name is Andy Garza,
And I'm Ana Martinez.
and we're going to be doing a presentation on adaptive immunity. So, what is an antigen? An antigen is essentially a molecule or molecular structure usually present on the surface of pathogens or foreign substances, which can trigger an immune response. An epitope, however, is a particular area of an antigen that immune cells or antibodies can recognize and bind to. The precise recognition sites, known as epitopes, are what set off the immune system's reaction to neutralize or eradicate the antigen. More so, B cell receptors, otherwise known as BCRs, are used by B cells to identify complete antigens, which are usually macromolecules, like proteins and polysaccharides. Additionally, major histocompatibility complex MHC molecules enable T cells fitted with T cell receptors, TCRs, to identify processed antigen fragments presented by antigen-presenting cells, otherwise known as APCs. T cells seek out intracellular pathogens by identifying processed antigen fragments, whereas B cells react mainly to antigens on the surface of pathogens. Because of this difference, the immune system can be quite resilient in defending the body against a wide variety of threats. Types of MHC and their roles in the immune system. It should also be noted that there are also two primary types of molecules that make up the major histocompatibility complex MHC. All nucleated cells have class 1 MHC, which is responsible for presenting endogenous antigens from intracellular pathogens to cytotoxic T cells. Helper T cells are presented with exogenous antigens from extracellular sources by MHC class 2, expressed on professional antigen-presenting cells. Because of these MHC classes present antigens through different processing pathways. They are crucial in coordinating immune responses. So, what is the role of dendritic cells in capturing and presenting antigen to T cells? Also, please be advised, desertic was a typo for dendritic. So, nonetheless, moreover, dendritic cells use a variety of receptors to facilitate effective antigen uptake. They are also essential for presenting antigens to T cells. Dendritic cells can internalize foreign antigens through cross-presentation, presenting them on MHC class 1 molecules to activate cytotoxic T cells against malignant or infected cells. Dendritic cells play a key role in immune surveillance by presenting and processing antigens to trigger specific immune responses.
Now, where does the development of lymphocytes begin? Now, transferring over to primary lymphoid organs, which consist of the thymus and bone marrow. This is where lymphocyte development begins. More so, while T cells go through maturation and selection processes in the thymus, precursor cells differentiate to become B cells in the bone marrow. To prepare lymphocytes for their roles in the immune system, these processes also provide them with the receptors and functionalities they need to identify and react to antigens. What type of selections are taking place during different stages of maturation? More so, different selections take place at different immune system maturation stages to guarantee the production of cell-tolerant and functional immune cells. T cells go through a process called positive selection in the thymus. This is essentially where those are able to identify cell antigens presented by thymic epithelial cells survive. What is the role of negative selection during the maturation process? Negative selection also stops the development of autoreactive T cells by removing T cells that respond strongly against self-antigens. These processes of selection play a pivotal role in molding a heterogeneous but self-adjusting pool of T cells, thereby enhancing the immune system's capacity to react to infections while bypassing negative impacts on the body's own tissues. Furthermore, a critical stage in the maturation of immune cells is negative selection, which makes sure that T cells expressing self-reactive receptors are either eliminated or rendered inoperable. Thymic epithelial cells present self-antigens to developing T cells in the thymus, where interactions between the two occur. Moreover, T cells, which have a high ability to react against self-antigens, are either eliminated via apoptosis or through functional inactivation, which essentially stops T cells from emerging and possibly triggering autoimmune reactions in the body. How and why is the vast diversity of receptor structures in the lymphocyte repertoire generated? Lastly, through a process called VDJ recombination, the lymphocyte repertoire's enormous diversity of receptor structures can be produced. Gene segments encoding the variable regions of B and T lymphocytes receptors undergo random rearranging during this genetic recombination, which process during their development. Through producing a wide variety of receptor structures by the immune system, lymphocytes can identify and react to a broad spectrum of antigens, which increases the efficiency of the immune response. Thank you.